everything that we have been talking about so far has been for one purpose: to get that image to be properly put onto the retina. How many times have I said that now? 1,000. The retina has all the beautiful, fantastic photoreceptors for which we can pick up that light, send it back to the brain to be interpreted. The retina itself is basically an extension of the brain. Okay? That's why those optic nerves come off of the brain there and travel on the underneath surface and attach to the eyeball so well. Okay? It is just an extension of it. We have the area of greatest visual acuity, the fovea centralis. Okay. Again, that's where we have 100% cones, okay, in that fovea. As you can see around it, I have a region referred to as the macula lutea. And this is where we have, again, a higher concentration of cones. Not necessarily 100%, but pretty dang close, right? And that's this region right here, okay? That's that region right there. Over here, we have our optic disc. You know that as the blind spot. As you can see from the picture here, it doesn't really do it spelling justice. There it goes. That's where all my arteries and veins kind of come in at. All right. There are no photoreceptors there, hence the blind spot. Okay. And I believe one of the labs looks at that concept. Okay. But you can kind of see there's that little sun I was talking about. Okay. And it actually has a cup to a disc ratio. That disc looks like a sunburst, right? And you should have a fabulous little cup to disc ratio there, okay? Again, if all you see is just the cup and no sun bursting around it, and if you look online, you could probably see some better images than this when it comes to detail on the cup to disc ratio. If all you see is just one big bright sun with no sunburst around it, then you know you're looking at somebody with high blood pressure or some kind of glaucoma issue going on there, some kind of compression going on. Okay. As you see these blood vessels here, oh my gosh, they're so beautiful. They're just smooth as they traverse through there. If you're looking at this eyeball and these things are very, tortuous in their movements, they're and then okay, then that tells you that there's too much pressure within there. And that's usually indicative of high blood pressure. Okay? And again, that's the stuff that they're looking at when they're trying to figure out the health of the retina, the health of the eyeball, and what they're looking at for some types of Alzheimer's diagnoses. So pretty interesting. Just looking at the eye, okay? Just looking at that. And you can see that with a traditional ophthalmoscope. You don't have to have a fancy pants computer. You can see that pretty decently with an ophthalmoscope. Again, all the magic is right here in the retina. And as you can see in the retina, we have kind of two major layers. And honestly, we're going to be spending all of our time in the neural layer, but there's also the pigmented layer. Okay. It does have a function. It does have a reason to be there, and we'll mention it. Okay. So I'd be remiss if we didn't talk about it. But as you can see in this neural layer, I have my rods and my cones. Okay. And this is how light is coming in. Okay. Coming in this way. So as you can see, light is coming in. So it's going through all of this nervous tissue. We have horizontal cells. We have bipolar cells. We have amacrine cells. And then ultimately, ganglion cells give rise to the fibers that make up the optic nerve. But these rods and cones, these are going to be my photoreceptors. Okay? These are the dudes that are going to be involved in picking up that light. Then we just go through a relay of various different cells here until we get to the ganglion. These cells here, we'll briefly mention a little bit about what they do, but oh my God, I could spend like 4 hours just talking about these stupid things. Okay. They give a ton of specificity. Okay. They will enhance your perception of it. They will create a nice solid image, right? When you're looking directly at something, it's, oh my gosh, I'm looking at that red of the bar there and the layers of the retina. Oh my gosh, it's coming in so crisp. As you get further away from the word the, it starts to become a little bit blurry. That's because some of these horizontal cells and endocrine cells actually will hyperpolarize. Okay, the light is coming in specifically right here and triggering these guys. Well, these over here will start to be inhibited. And that's where some of these other amacrine cells and horizontal cells come into play. Okay? It just helps to produce a nice, crisp image. I'm not going to get into all that mental gymnastics because it is honestly maddening. Having us just understand how rods and cones work, that's what I want you to really know. Okay, so we may mention a little bit about the horizontal cells and the amacrine cells, but we're not going to do a deep dive into those. We don't really need to. Okay, picking up vision, picking up that light so that we can see stuff. That's really what I want you to know. Okay, so if the book starts going off on tangents with the amacrine cells and the horizontal cells, don't worry about it. Okay, don't worry about it. My photoreceptors are in the innermost part of that retina. Rods and cones will synapse on bipolar cells. And what did we say about bipolar cells? They're always associated with special senses. Here they are. So I go from my rod, I'm just going to put an R, and my cone, we're going to synapse on the bipolar cell. Okay. And that will synapse on a ganglion cell and go to the optic nerve. Optic nerve. Okay. So we have rods and cones. I have bipolar cells, bipolar cells, bipole C, and then we have ganglion cells. Okay, that is basically the gist of what we're looking at. Ganglion cells, ganglion C. Okay, again, amacrine cells, horizontal cells, they're just going to be there to help ensure a nice, crisp, proper image. When we look at rods and cones, they're named because they're shape. That's it, right? Cones have this conical shaped tip, and rods have a rod shaped tip. I mean, that's as easily as something can be named, right? Except maybe cuboidal or columnar. So these are our dudes that are detecting those lights. You can see here we have an outer segment, okay, and an inner segment. The inner segment, guys, all it is just a typical cell. Okay, it has the nuclei, it has the organelles, it has all the doodads that's going to maintain the integrity of the rod and the cone. It's this outer segment that has all those photosynthetic pigments in it. Rhodopsin, all the photopigments that we're not going to be mentioning, but we'll briefly talk about. Okay, this is where all of our photopigments are found. Right. So obviously this is going to be the region This is going to be the region right here in this outer segment where all the picking up of those photos, those photons, the visual light spectrum is going to be occurring. Okay? This is where the magic happens, out here in these outer segments. This is going to create that graded potential, that receptor potential, and send it down the length of this photoreceptor, triggering the bipolar cells, horizontal cells, okay? Ultimately to the ganglion, ultimately to the optic nerve. Okay? Then here, much more kind of rodish and conical type shape. Okay? But rods and cones. Okay? Rods are the ones that are associated with grays, blacks, and whites. Dark vision. Cones are going to be the ones for color. Okay? They're going to be the ones that give the greatest visual acuity, ultimately. Again, we talked about two layers. Okay, the retinal layer, I'm sorry, the neural layer here is where all the action is going to be ultimately occurring and happening. The pigmented layer does have some importance. Really, single layer of cells, boring, located under the rods and cones. Okay, sure. But it does a lot of fantastic things. As the outer disc has some wear and tear from the picking up of photons of light, okay, that's going to cause some wear and tear, and it's just going to kind of phagocytize off. So it helps to clean up the shrapnel that occurs with cellular wear and tear. It absorbs excess light. Okay, okay, sure. But delivering nutrients is a little bit more important, at least, comparably speaking. It's closer to the choroid, so it's going to have a much more closer access to nutrients, which can exchange it with the rods and the cones. Okay, which again, these are cells. These are cells. Okay, and they obviously are undergoing metabolism, producing waste. Producing waste, so we need to exchange it and get rid of it. We need to bring the good stuff in. It does contain this ability called a blood-eye barrier. Blood-eye barrier. And we've talked about the blood-brain barrier. It's the same thing. Okay. There's also a blood-eye barrier. It prevents immune cells from coming in there and attacking the retina. That wouldn't be very conducive, right? We see the same phenomena in a number of locales, not just the brain. Here, we see in the testes, we have a testis blood barrier. I mean, sperm is, we created, but the body doesn't necessarily like the cell. Doesn't necessarily it's not supposed to be there. So that testes barrier does the same thing. Suppresses an immune attack, which is good. Sperm can survive, species can live on. It will also participate in the visual cycle of the retina. What in the hell does that mean? That means the recycling of vitamin A, ultimately. Vitamin A is going to be very important in helping us to create the photopigments. So it's going to help with the recycling of the photopigments, getting vitamin A in there to incorporate so I can make more photopigments. Because as soon as light hits these rods and cones, boom. Okay, photopigments are changing shape and being broken down. We can regenerate, obviously. It takes some time. This helps deliver the goods, vitamin A specifically, to help us to recycle and create more photopigments.
What's it mean by shed outer discs?
As the wear and tear occurs here, okay, these will slough off. The ends of these will slough off.
So it's just the very ends of the
Yep.
rods and cones.
That's all that happens. I mean, it's just normal cellular wear and tear that happens with any cell. Okay. And we'll just kind of make more. Okay. We'll make more here. This will shed off. Part of this will kind of shed off here at the tip. And it we'll just continue to, grow more outer segments there, outer portions. And that's all that means. Because if all that shrapnel from wear and tear was allowed to accumulate, obviously that would, potentially affect the functionality of those rods and cones because it would interfere with light coming in eventually. So it helps us just to get rid of that. I mean, there's a certain pigment. Not only is it a blood-eye barrier to suppress immune attacks, it also creates a nice stable environment for the photopigment, or sorry, for the photoreceptors to work properly. So it maintains specific ionic concentrations. So it does a lot of stuff. But in terms of vision, in terms of getting those photons of light to hit the rods and cones and then the magic to happen, that's what I'm more concerned about. So we're talking about the neural layer a heck of a lot more than this pigmented layer. This is the only spot that we'll talk about it. So obviously it's not going to be short answer. Multiple choice is where you're going to see me asking questions about this sucker because I'm not going to talk about it in terms of vision. So this leads us into looking at these rods and cones. I think I mentioned it earlier. Rods see in black and white. Okay. These are your low light photoreceptors. Okay. So when you're in a dark room, these are the individuals that are going to be kicking into gear to allow you to see within it. Okay. The photopigment for rods is rhodopsin. And we get this from basically vitamin A. Okay. When light hits the rods and the cones, it will cause the photopigment, rhodopsin, to disassociate. And I have a picture somewhere dealing with that phenomena, what that looks like. Okay. It's not very spectacular. Rhodopsin will disassociate. It breaks apart into its component parts. Okay. Retinaldehyde, which is my vitamin A, and opsin, the pigment. Okay. When rhodopsin removes itself from the rod, it is referred to as bleaching. Okay. The photoreceptor bleaches, okay, because it doesn't have its pigment within it anymore. And in about a minute is all it takes for this photopigment to disassociate. What does that look like? Okay, here's my rod. There's rhodopsin. When light hits, you can see it. It changes shape. It's in the cis form when nothing is hitting it, no light. And as you can see, as light hits this photopigment, you can see it changes shape. It goes from a cis shape to a trans shape. 11 cis, whatever. I don't really care about that. Okay. Understanding that it goes from a cis to a trans changes its shape. Yeah, that's more important to know. I don't care 11 cis, to be honest. All right. But as light waves hit, in about a minute, this thing completely disassociates from the photoreceptor. This is the bleaching effect. Okay. Here you can see it completely disassociates from the photoreceptor. And we have to bend it back. Okay. We have to get it back to the cis shape. This is where vitamin A comes into effect here. It allows this to be recycled, so it changes its shape back. We can associate it back into the photoreceptor, and it's ready for action again. So rhodopsin is in a cis form. Okay, it has a specific shape. That's all I'm trying to get across. After the light hits it, after about a minute or so, okay, it bleaches. That light causes the rhodopsin to change its shape. And it changes it into a trans form. Okay, so we are completely changing the shape of this thing because of light hitting it, but that's okay. It will remove itself from the photopigment, from the photoreceptor, okay, and it has to be converted back, and it does. Okay, it does not stay disassociated for an extended period of time. Okay, well, I guess it can. If it's just normal light, it disassociates. And it takes a few minutes and it reincorporates back and it turns back into the cis form and it's ready to do it again. Okay. And again, this recycling of the photopigments is a function of that pigmented layer because that's closest to the blood, which is closest to the nutrient supply for vitamin A. All right. Sorry, some in normal light, like it is right now, okay, this is not a huge thing. If I go from a completely dark room and turn the lights on or go from somewhere dark and then walk outside into the light, that is above and beyond. Okay, that's not normal light. Okay, you're going from one extreme to the other. You're gonna cook all of that rhodopsin. Okay, you're gonna bleach the heck out of it. You're gonna burn through all of your rhodopsin stores. Okay, that's why it's very difficult to see when you come out of a movie theater when it's pitch black like that. All right, very dark except for the light from the screen, but really, really, really dark. And you walk out into the light, the ability to see all that light coming in. It's your eyes are really sensitive, okay, because it's geared for dark vision. Okay? It's called dark current. Sounds mysterious when you say it like that. Your eyes are running on what's called a dark current. It's functioning in minimal to no light. So it's only your rods. And when the rods are still doing that and you walk out that door of that movie theater and all of a sudden it's nothing but light, all those rods are activated and they get toasted. And your ability to see goes right down the pooper. Okay? Because rods don't function in bright light. You've burnt through all the rhodopsin, all those photopigments. Okay? And it takes fractions of a second to get those cones to kick into gear and start to see with some great visual acuity, right? It takes a handful of seconds. Depending upon your diet, your age, depends on how quickly you're able to increase your visual acuity, from going from a completely dark to a completely bright type of state. But all that happens because we bleached it. And a few other pieces of magic that we'll talk about here. So here is this dark vision, okay, this dark adaptation, okay? You can see it takes about 20 minutes to completely adapt to the dark. So you go from a bright, you go into the dark, the same thing happens, okay? Your ability to see in huge amount of detail is dramatically less. It only, guys, I mean, this says 20 minutes, but in all honesty, it takes about 5 minutes. 5 minutes to regenerate half of the rhodopsin. Okay. So if you've bleached it all out when you've been outside looking around, okay, you've bleached it all, right? And honestly, rods are hyperpolarized in light, so they're not sending any images back. But you're burning that rhodopsin, you're burning that photopigment, light's still hitting it. So it only takes about 5 minutes to regenerate. Takes about 90 seconds. 90 seconds for the cones to regenerate. Half. So 5 minutes, 5 minutes to regenerate half the rhodopsin. Takes 90 seconds to regenerate the cones, photopigments. Okay. That's G, photopigment. About 20 minutes in, it's dang near 100%. To fully regenerate, though, takes about a half hour. Okay. So it does take some time. I probably won't ask the times at all. It's just there to kind of give you a frame of reference. Okay. Because we're going to be talking about not being able to see and whatnot. And that sounds like it takes forever to come back. And it doesn't. It takes a handful of seconds. Okay. It takes a handful of seconds. All right. So here is the electrical activity of rods. I don't know why they put retinal cells. This is rods. Okay. Rods work in the dark. So we are going to be talking about rods here. Okay. We are back to sodium. We are back to sodium being a depolarizer. The only time it's not is for the ear. Okay. The only time it's not a depolarizer, it's going to be in the ear. Everything else, the depolarizer is always sodium. Okay. That's the one issue that people screw up on an exam. Okay. Sodium is what's going to depolarize. When it becomes dark, as you can kind of see here, this is where we can, depending on the specific type of cell, depolarize or hyperpolarize this thing. Rods are going to be our photoreceptor of choice when we are in a dark environment. So when we are in a dark environment, our rods are kicked in. All right. So here is, on the left, the dark pigment. Okay. Here's another image of this kind of as well. Okay. Here's a rod in dark. A rod in dark, well, yeah, there's my photopigment right there within the rod. Okay. As you can kind of see, this is a secondary messenger system here. Okay. Light hits my photopigment, and we get retinol to change its shape from a cis to a trans. We bleach it. When this happens here, okay, in dark, there's no light. I have my photopigment here. Okay. I have my sodium gated channels here. And as you can see, they're open. Rods function in dark. Rods function in dark. So as soon as you go into a dark room, the rods are what are producing signals. Sodium channels are here. Okay. They're open. There's a high degree of cyclic GMP in there. That is helping to open up these sodium gated channels. Okay. GMP is the specific secondary messenger here, and it's what's keeping this sodium channels open. Sodium rushes in, and that causes depolarization. Again, this is in dark. As the wave of depolarization, this receptor, okay, potential travels across. Okay, this is a very short distance that this thing is traveling. As this wave of little bit of depolarization occurs, excuse me, voltage gated calcium channels open up, like in any nerve. Synaptic vesicle causes it to interact with the V snares and T snares of the vesicle and of the membrane here, interact and exocytose the neurotransmitter out. And that's going to synapse ultimately on this bipolar cell. When light hits the rod, okay, when light hits the rod, and I'm still in dark, okay, even if I shut off all the lights, I still have light emanating from my computer screen. Okay, that's the light. When I'm in a dark room and that light hits my eyeball, that's when we activate rhodopsin. Completely dark, no light at all, right there. Nothing's hitting it. Okay, there's nothing to bleach it. As soon as that light gets produced in a dark environment, I change the shape of rhodopsin from cis to trans. This action right here will lead to that bleaching, right, in about a minute. Okay, if it's less than a minute, okay, it recycles itself right there. It will get bent. Okay, but after about a minute, this is going to dissociate and bleach. As soon as this sucker hits it, right, this is when we kickstart a secondary messenger system here. Okay, I don't care what the second. I'm not going to ask the secondary messenger. Okay, we kick in a secondary messenger system. This activates cyclic GMP, which is going to cause it to be broken down. When I start to break down cyclic GMP, there's not as much cyclic GMP to bind to the specific receptor now to allow sodium to come in. So if sodium doesn't come in, I'm not depolarizing. It stays hyperpolarized. When we hyperpolarize, we make it more negative. Okay. The ability to send an electric potential diminishes. I only have a tiny little bit of sodium coming in. So that corresponds with a tiny amount of neurotransmitter being released. And that allows me to see an image, but not with great visual acuity. If I want to see in great detail, I need light. Okay. But in a dark environment where rods are king, light actually inhibits their activity. So when you're in a dark room, they are sending off potentials, graded potential, which gets to the ganglion and ultimately becomes a action potential. Okay. We are firing off graded potentials here, a receptor potential. That's allowing us to interpret what we see around us, even though we have no light. Okay, you've been in a dark room before. Okay, you may not be able to see very well, but after a few minutes, you can start to make out some things. As soon as you're in that dark room and a light starts to emanate from something, now we start activating that photopigment. Okay, rhodopsin goes from a cis to a trans shape. That is what kickstarts the secondary messenger system. And we basically break down cyclic GMP. By breaking that down in a dark environment, in the presence of light, I actually close some of these cyclic GMP channels, which allow sodium to come in. Sodium is my great depolarizer. So with less sodium coming in, I don't depolarize, all right? I stay hyperpolarized, or I get a little bit more negative. As I'm hyperpolarizing, I am not releasing as much neurotransmitters. Travel across the synapse to bind with that bipolar cell. Okay? So the number of receptor potentials diminish drastically. If I have fewer receptor potentials going to the bipolar cell, to the ganglion cell, ultimately to the optic nerve, if I have less action potentials getting back there to the optic nerve, to the occipital lobe, I'm interpreting that as I'm in a dark environment, okay? And I'm seeing, not in great huge detail, but I know there's something kind of over there on the floor. And if I go that way, I may trip and fall, or it may bite me. I don't know what the hell it is. I can't make it out very well. There's not much light. Okay. What's going to give me all that great vision acuity, though, is a cone. If I can increase the light, then I'm not going to be worried about rods, but now I'm going to have cones kick in. And cones are going to give me that good, great visual acuity, right? Because they're active in light. As you can see, rods aren't active in light. They're hyperpolarizing. Okay? So they decrease their action on a bipolar cell, which decreases action on a ganglion cell, which decreases action sending back on that optic nerve to the occipital lobe. Okay? Everybody's still with me there. When it comes to vision, it's a little bit weird. Okay? Cones have a number of photopigments, and I believe, yeah, photopsins is what I have, just a general term. Okay. There's different ones. There's blue, there's green, there's red. Those are the big three when it comes to various different types of colors here. Okay. just detecting various different types of wavelengths within the color portion of the spectrum here. For cones to kick in, I need some light. When it comes to the number of cones active, I don't necessarily need as many of them. So cones are less sensitive. Okay, they don't burn out. Right? They need the light hitting them to function properly. Okay? So they are opposite when it comes to how they depolarize or hyperpolarize, comparably to okay, when it comes to rods. When the light hits them, they do depolarize. Okay, they do depolarize. When there's no light, they are hyperpolarized and they are not active at all. Okay. So when it comes to photoreceptors being triggered, that's the main difference. Okay. In a dark environment, it is rods. In a light environment, it is cones. For cones, in light, depolarize, and depending upon the wavelength of that light medium, different types of photopigments are going to be more active than others to absorb these different wavelengths of light. I don't know why my thing's not working. Okay. So if it's in this wavelength, it's your blue cones that are going to be more activated. If it's in this spectrum here, you can obviously more green, and you can see there's a good deal of overlap. Yellow and green and red and blue and green. I mean, you could see we can get all the colors of the rainbow here by just these three little dudes here. Okay. So depending upon what the wavelength of that vision spectrum is, of that light coming in, will trigger different cones. And you can see I have some variation that can occur here. I can trigger more than one. Okay. So I could trigger all three at some points. And this allows me to see in that color. Again, where I try to focus light by either moving my head, moving whatever it is I'm reading, I try to get it on that fovea centralis. Macula lutea, fovea centralis. I try to get on the fovea. Okay. That is where my greatest visual acuity is. That's where it's predominantly all cones. Okay. All cones. Okay. And there's a relationship, and I'm not going to ask what the relationship is, but that's what they're trying to show you here. Okay. One cone with one ganglion cell. Okay. One cone with one ganglion cell. That's what they're trying to show off here. Right. And when you do that imaging study at the ophthalmologist office, ophthalmologist office now, I mean, you can you can see this visible pit. It's kind of like an MRI, and how they can kind of go deep and shallow when they're going over the image study with you. It's pretty cool. But you generally have a 1-to-1 ratio there within that pit, within that fovea. And again, that's going to allow a huge amount of visual acuity because of that. As you get further away of that, you can kind of see it will, vary a little bit as we get further away from that fovea centralis, in around 100-to-1. I'm not going to ask the number. Okay, it's just there to show you that great vision acuity is there, because for every one cone, we have one ganglion cell associated with it for the most part. So that's why we see with great visual acuity. It's firing off. They're all firing off, all sending an image. by allowing vasodilation to occur, vasodilation, dilation of the pupil to occur, I can allow more light to come in. When I allow more light to come in, I can stimulate more rods in a dark environment. Okay. So this allows a heck of a lot more rod activity to occur. When it's light outside, and there's a good deal of light outside, like it is right now, that pupil constricts and it limits the amount of light that comes in. That's okay. My cones are very sensitive to light, right? So I don't need to blast my entire back of my retina with light to get them to fire with great visual, to provide great visual acuity. When I'm in a dark environment, I need to open up those suckers. Right? I need to bring in as much light as possible so I can make out an image. And again, it's that depolarization event or that hyperpolarization event that occurs in the darkness that the brain is going to interpret. So even though there's not as many impulses going back to the occipital lobe there, where the vision center is, we're going to interpret that as I'm in a very dark environment. And if only rods are active, they only give so much visual acuity. As our eyes, as the saccadic eye movement occurs, as you're normally looking at something, your eyes are constantly kind of doing these small little bitty movements, okay? You're not actively doing it, okay? You're not saying, look this way, look that way, look down, look up, look. You're not actively doing that. These are just constant small little shifts in a person's eye. And if you pay attention to somebody when they know you're not looking at them, when you look at the eye, you'll see small little shifts that occur. Okay, this allows them to bring information from all parts of that visual field. Okay, so I mean, I know what's going on over here. Okay? I know what's going on right over here. I'm not looking straight at it. I'm looking right. I'm looking at you, but my eyes are totally shifting all the time. Small little movements. Okay. And that's that saccadic eye movement. So we're bringing in information from all parts of the visual field here. So even if there's a little bit of light right here, some of it's still going to get funneled into the eyeball there and allow me to make out what that is. In light, when my cones are active and I have great visual acuity and I'm looking straight ahead, I can see something fuzzy, moving over here. Again, light's coming in. If it was pitch black outside and I had that same thing going on, where somebody was walking down the sidewalk here, I wouldn't necessarily see them because my rods don't have great visual acuity and they're going to be hyperpolarized. Okay, so they're not going to be sending as many impulses. So it's all about light. It's all about light. How much is there? Is there none? Dark current. Rods are going to be activated. If it's in light, cones. And again, sodium is going to be my great depolarizer. Okay. When it comes to rods and cones, again, I keep saying it doesn't take as many cones, and your eye does not have as many. Okay. It has a hell of a lot more rods. I think the book says 120 million rods, and there's only 60 million cones. So that's what, 126 million little nerve fibers extending back. But again, remember there's some ratios there. So there's only like 1.2 million axons that extend and form the optic nerve. So again, it doesn't take a whole lot of rods to be stimulated to cause great visual acuity. For the most part, this is the system here that we use. This is the system that, if I was to ask, how do we interpret photons of light in the eyeball? This is the pathway. That optic nerve extends back to the thalamus, basically. Okay? From the thalamus on back to the vision cortex and the occipital lobe is right here, is that optic radiation. That optic radiation is just the branching of different pathways that extend from the thalamus back toward the occipital lobe, where the vision cortex is. So the ganglion cells are what form the optic nerve. The optic nerves cross over at the optic chiasm. We talked about that with the vision fields. The optic nerves extend back towards the thalamus. And it'll go to, superior colliculus, where we have reflexes, goes to the lateral geniculate nucleus. I'm not going to ask about the LGN there. Okay. From the ganglion cells to the optic nerve, from the optic nerve to the thalamus. And remember, most everything is going through the thalamus, right? Except for smell. From the thalamus, we extend back to the primary vision cortex, which is in the occipital lobe. And this is how we figure out what in the hell we're seeing. The superior colliculus, again, is more reflexive, saccadic eye movement, just natural reflexes that occur. And if the geniculostriae system is 80%, the tectal system is about 20%. Okay? I'm not going to ask you, I will not ask you what the tectal system is. I will not ask you what the geniculostriae system is. If I ask for a specific pathway, you're going to tell me ganglion cells, optic nerve. Optic nerve crosses over at the optic chiasm. And there's the optic tracts. Whether you say optic nerve or optic tract, I want you to tell me that it's ending into the thalamus. There we can synapse on a few different things. From that thalamus, though, I relay the information back to the occipital lobe. That's where my primary vision cortex is. And that's where I figure out what the heck I'm looking at. Reflexively is the superior colliculus. And thalamus can send some relays there. And these are just, as soon as you see something move in your visual field, it's a reflex. Your eyes immediately kind of go to it. Okay, it always happens. And that's the tectal system. But I'm not going to ask how much percentage and what comprises the tectal system. What's a reflex for vision? Superior colliculus. And that was a structure that we mentioned when we looked at the midbrain. Superior colliculus, inferior colliculus. Inferior, superior. Okay. Inferior colliculus, reflex for hearing sounds. As soon as you hear a sound, you're going to localize it and look in that direction. The tectal system, when something starts to move in your field of view, you're automatically going to look and see what the hell that is. Okay, to see if it's something that's going to affect you or not. And ultimately, where do we send the information to interpret what we are seeing? Primary vision cortex, the occipital lobe. Yes, exactly. That's what I want you to know.
It's like 98% of the time, almost all of this stuff is going to be sent there. It's just the pathway might differ slight. But even then, the biggest thing that you really want us to know is that wherever it may come from, it eventually goes to the thalamus, which eventually just ends up in the occipital lobe.
Yes, sir. Yes. Because again, I'm going to ask one of these special senses as a short answer essay question, however you want to say it. Okay. I'm going to ask, describe how sound waves are traveling from this to be interpreted. So you got to go through the outer ear, middle ear, into the inner ear. Describe what the business is in that inner ear, okay? In the cochlea, right? The nerve that it travels, where it goes to, where it's interpreted, temporal. Photon of light, follow a photon of light to be picked up and to be interpreted, okay? You'll go through the tunics, go through the structures of the eye into that retina. Is it in light? Is it in darkness? Who's going to be engaged? Darkness rods, light cones. Where does it travel? Ganglion cells, well, photoreceptor, bipolar, ganglion cells, optic nerve, back to the thalamus, and ultimately to the vision cortex, occipital lobe. That figures out what we see. The other thing is superior colliculus, okay? And superior colliculus is always reflexive for the eyes. As soon as you see something coming in your field of view towards your head, you reflexively move it, okay? Or if you see something moving off in your field of view, you look, okay, I see it, okay? It allows you, this tactile system, the superior colliculus, is what allows you to figure out where the object is in your vision field. So I'm going to ask one of these. Be balance and equilibrium. Hearing. Vision. Those are the big three. I'll probably ask one of those as an essay question. A lot of moving parts. So that obviously makes a good short answer question. I'm just going to